Hello dear students. The topic for this class is complement system. So it's again a part of immunology. So the important questions from this part that can be asked is classical pathway, alternate pathway, and sometimes deficiencies of complement can also be asked. But we have to be familiar with the pathways of complement system, that is more important. So first of all, we'll see what is meant by complement system. Complement system, or you can call it as a complement cascade. Actually, these are some proteins which are present in serum as part of innate immunity. It's present in our serum in inactive form. When they get activated, they augment the immune response. So they help in clearing the microbes from the serum. They help in removing certain infected cells, destroying cells. So they augment the immune response. Not only that, they can enhance phagocytosis, they can cause anaphylactic reactions, inflammation. So there are various applications of the complement proteins, but basically they augment the immune response once they get activated. So they are serum proteins which are present as part of innate immunity, which is present in the serum in an inactive form. Once they get activated, they augment the immune response. So what are the components of complement? You have 9 components of complements or complement proteins from C1 to C9. You can call it as C1, C2, like that up to C9. So they get activated in a cascade pattern. That is, once one get activated, it will activate the next one. That will go and activate the next one. So that's why it's called a cascade. And in the C1 to C9, for C1, we have 3 protein subunits, that is C1, Q, R, and S. It is the C1q that gets bound to this antigen antibody. That is the first molecule that gets activated. That is the binding component of complement, which will in turn go and activate its other subunits R and S, which is having an enzymatic activity. That will cleave the other complement protein and will trigger the cascade or trigger the pathway. So C1 to C9 are the basic components, and C1 has got Q, R, and S. And we have 2 major pathways for complement: classical pathway and alternative pathway. In addition to classical and alternative pathway, we have another pathway for complement that's called the lectin pathway. Lectin pathway, which is not that important. What you have to learn in detail is classical and complement pathway. So when these pathways get activated, what they do is they will finally cause lysis or damage of the target cell. So whichever cell is the target for the complement pathway, complement system, they will finally lyse the cell and destroy it. That is their target action. So for any complement cascade, whether it be classical pathway or alternate pathway, there are 4 steps. First one is initiation of the pathway. The pathway has to get activated, it has to get started. Initially, it will be in inactive form, it has to get started. So for that, it needs some initiators or triggers. So that is the initiation of pathway. That is the first one. Then there will be a series of cascade leading to formation of C3 convertase. C3 convertase is an enzyme, is a complex which has an enzymatic action that will activate C3. So this is a very important step, formation of C3 convertase. After that, the C3 will activate C3. C3 convertase will activate C3, which will join the cascade and form C5 convertase. So that is the third step, which is very important, which in turn will go and activate C5. So C5 convertase is responsible for activating the C5 complement protein. And finally, all these will end up in formation of membrane attack complex. So once C3 convertase is formed, it then forms C5 convertase, and finally everything will lead on to formation of a membrane attack complex. You can call it as MAC, membrane attack complex. So that will cause the lysis of the target cells. So these are the 4 important steps in any complement cascade. So in classical complement pathway, I told you the first molecule to get activated is C1, C1QRS. Among QRS, Q is the first one that is getting activated, and then the next one is 4, then comes 2. So when you write 1 to 9, the changes after 1, it is the 4 that is getting activated. So then comes 2, 3. So 4 is already activated. So then comes 5, 6, 7, 8 and 9. So this is the sequence of activation of complement cascade in classical complement pathway. See, 5 to 9 molecules together forms the membrane attack complex. So we'll see the classical complement pathway, how the cascade progresses. So before that, you have to know what is the initiator or trigger for the classical complement pathway. So the most important trigger or initiator of classical complement pathway is the antigen antibody complex formation. So here we are seeing like E plus A. Why? Because complement pathways were studied on RBC lysis mediated by complement. So E for erythrocyte and A for the anti-erythrocyte antibody. So if an erythrocyte and anti-erythrocyte antibody binds, it will trigger the complement pathway, which can finally lead to RBC lysis. So that was how these pathways were studied. So that is why it is depicted as E plus A. You can write it as antigen antibody complex. That will be better. Antigen antibody complex. So the first molecule to get activated is the C1 molecule. So I told you C1 has got C1q, R and S. So it is the C1q that gets bound to antigen antibody complex. And what happens when C1q binds is C1q will initiate, when it gets activated, it will initiate C1R and C1S. C1R and S also gets activated. Actually, C1S is having the esterase activity, which can cleave the next complement, that is C4. So the first cascade, the initiator antigen antibody binds to C1, C1QRS, and the next thing that is coming into action is the C4. So C4 is cleaved by this complex initiator. In the complex which is formed initially, C4 is cleaved. C4 gets cleaved into C4A and C4B. So most of the complements, when they gets activated, they gets cleaved into the A and B components, of which mostly B remains in the cascade and A is removed. And most time A has got an anaphylactic action, anaphylatoxin. They behave as anaphylatoxin, so they can initiate inflammation, the A components. So in this case, after C1, it is the C4 that is getting activated. C4 is cleaved into C4A and C4B, and the C4B joins the cascade. So we have the antigen antibody complex with C1. So along with C1, the next thing that is joining the cascade is C4B. So C4B is joining the cascade, and so after C4B, what happens? See, so now it is the antigen antibody C1 4B. It is the next complex that is formed. Now this, in presence of magnesium, it will cleave C2 into 2A and 2B. The only exception for what I mentioned before is C2. In case of C2, it is the 2A component that is joining the cascade and 2B is being removed. For all other components, it is the A component that is removed and B component that is joining, except for C2 where the B removes and A gets joined into the cascade. So now the C1, C4B becomes C1, C4, C2A. So now we have the antigen antibody complex along with C1, 4B, 2A. Actually, this complex behaves as the C3 convertase. This has got an enzymatic action which can cleave C3. So we have come to the next major step in the cascade, that is formation of C3 convertase. So C3 convertase is C1, C4B, C2A. C1, 4B, 2A. Now this behave as C3 convertase, and C3 is split into C3A and B. Remember, A removes, B joins the cascade. So now the cascade becomes C1, 4B, 2A, 3B. 3A has been removed. 3A has got anaphylatoxic properties. So now you have come to the next important step, that is formation of C5 convertase. Okay, so 1, 4B, 2A, 3B is the C5 convertase. Now, what does C5 convertase do? C5 convertase will split C5 into C5A and C5B. Again, 5A is removed, it's an anaphylatoxin, and 5B joins the cascade. Now, this point, 6 and 7 will add on to 5. And C5, C6, 7 now again joins 8 and 9. So now it is from C5, when C5 is converted into 5A and 5B, C5B, that is the starting point for the formation of membrane attack complex. So to this, now you have the C3B, now C5B, and to this what happened to C5? the other components join 6 to 9 will join with C5 together forming the membrane attack complex. So the pathway which we mentioned from C1 to C9, after 1 it is 4, then comes 2, 3, 5, 6, 7, 8 and 9. The last part after C5 convertase formation, the last part is when 5 gets activated, it triggers the activation of all the other components like 6, 7, 8 and 9. Together they form the membrane attack complex, and this membrane attack complex will attack the membrane and it will cause lysis of the cell. So in this case, if it's an erythrocyte and RBC, finally it will lead to RBC lysis. So that is what is happening. So remember, the membrane attack complex is C5 to 9. And what is the C3 convertase in classical complement pathway? It is C1, 4B, 2A. That is the C3 convertase. And what is C5 convertase? C1, 4B, 2A and 3B is the C5 convertase. So that is the classical complement pathway. So next we are going to see the alternative complement pathway. So we should know what are the differences of alternative from classical complement pathway. Actually, classical complement pathway gets activated by binding of antigen and antibody. So they get activated only after the adaptive immunity comes into action. But alternative pathway doesn't need that because it gets activated by the presence of certain components in the microbes like the endotoxin or the lipopolysaccharide or the chymosan which is present in the yeast cell wall. These are the initiators of the alternative complement pathway. So what do they do is, in alternative pathway, there is no involvement of C1, there is no involvement of C4. These two components are lacking in alternative pathway. Instead, it is beginning with C3. And finally, it has to form the C3 convertase. Okay, so for that, initially C3b, which is present in circulation. Remember, C3b is present in circulation in a free form, which is kept inactivated by certain factors. Normally C3b doesn't get activated, but when C3b in circulation binds to endotoxin like molecules, they get activated. And what they do is they will bind to factor B. Factor B comes into the cascade. It's factor B or piperidine factor. And this factor B also needs to get cleaved into BA and BB. For that, factor D comes into action. So this B is split into BA and BB, and only the BB joins the cascade. So we have C3b, capital B, small b. Now this is the C3 convertase. So this has to again act on our normal C3 and split it into C3a and C3b. Now this C3b, capital B, small b, to this your C3b also joins. This together will form the C5 convertase. Okay, so initially it is starting with C3, C3b, but that's not enough. It has to finally go and cleave the normal C3. Then only it can form a C5 convertase. So C3b binding to endotoxin and adding on factor B, this factor B getting cleaved by factor D, and finally this is acting as a C3 convertase, and you have now C3b also onto the cascade. So 3b, capital B, small b, C3b. So on the beginning and the end you have C3b, in middle you have a capital B, small b. This is the C5 convertase of alternative pathway. So the rest of the cascade is the same as the classical complement pathway. Once 5 gets activated, 6, 7, 8, 9 also gets activated, joins the cascade and form the membrane attack complex. So rest all things are the same. Remember, the initiator is molecules like endotoxin and it is starting with C3b, and there is no involvement of C1 and C4 in alternative pathway. So in summary, classical pathway, alternative pathway. You can see classical pathway start with C1QRS, joining on to 4 and 2, coming to 3, and finally the 5 to 9 components. Alternative pathway, it is starting with C3b and factor B, forming a C3 convertase, and rest all things are the same. In addition, I told you there is one more pathway called lectin pathway. Lectin pathway, here some microbe associated proteases, certain microbial molecules bind to lectins in host cells. This combination will trigger the activation of C4. So there is no C1 involvement in lectin pathway. Rest all the things are same as classical pathway. The only difference is the initiator is binding of lectin with the microbe-associated serine protease. Certain microbial molecules bind to lectins in host cells, which will trigger the complement cascade starting from C4. So there is no C1 in lectin pathway. Even that is not that important for us. What is more important is classical and complement pathway. So remember one more thing, the anaphylotoxins, the C3A, C5A, C4A, all those things which are released into the serum initiates inflammation. And these components, the B components, they are very good opsonins which will trigger phagocytosis. So all these are actions of the complement molecules. So what are the effects of complement molecules? They can cause bacterial lysis, they can cause cell lysis, initiate inflammatory response by releasing these anaphylotoxins. They can trigger endotoxic shock, opsonization, and thus helps in improving phagocytosis. They can trigger the hypersensitivity reactions like the type 2 and type 3 reactions, and they can also help in autoimmune diseases. So complement, when it comes to play, it will cause cell lysis, it will try to remove the microbes. In addition, it will also stimulate certain other unwanted things like inflammation, or it can go trigger shock, or it can lead to autoimmune diseases and hypersensitivity reactions. So all these are the biological effects of complement pathways. Finally, deficiencies of complement. Complement deficiencies, one or two things you have to remember. Suppose you have a C1 inhibitor deficiency, which is keeping C1 action in check. If this inhibitor is deficient, C1 will always get activated, which will lead to enhanced complement cascade activation that can lead to enhanced inflammation and enhanced anaphylotoxin release, which will lead to a condition called hereditary angioneurotic edema. So hereditary angioneurotic edema is due to unwanted triggering of the complement cascade due to C1 inhibitor deficiency. Suppose you have the first three components of classical complement pathway missing, it will lead to these three components are missing in certain autoimmune conditions like SLE and other collagen vascular diseases. You know, SLE is an autoimmune disease, so there will be more of autoantibody formation, antigen antibody complex formation, which will consume the complements. Complement cascade will always get activated. So that can lead to deficiency of C1, 2, and 4 components. Now, C3 and its inactivator deficiency will lead to recurrent bacterial infections. If you have the membrane attack complex components lacking, it will also lead to increased bacterial infections, mainly gram-negative infections. And C9 has got, Elon doesn't have much problem, but if all the membrane attack complex components are lacking, there will be increased bacteremia due to gram-negative diplococci like Neisseria. So these are the deficiencies of the complement which leads to various conditions. So again, once more, the classical lectin pathway and alternate pathway. Classical pathway is C1 to C9. Alternate pathway starts from C3b. There is no 1 and 4 involvement. Lectin pathway starts from C4. There is no C1 involvement. And remember, the initiator of classical pathway is antigen antibody complex, whereas it is certain pathogen related molecules like endotoxin in case of alternative pathway. So that is about the complement system. Please learn the cascades well. Okay, the next topic is major histocompatibility complex. So this also you should be familiar with to know all the reactions taking place in the immune system. So what is MHC or major histocompatibility complex? The name indicates histocompatibility. So these molecules or these genes and the proteins they code are important in determining the tissue compatibility in case of a transplant or in case of a graft. So it determines whether a graft is accepted or rejected, because these antigens present in the cells are important in immune recognition. So actually MHC is a group of genes which code for proteins which are found on the surface of the cells that help the immune system to recognize foreign substances. So these are the molecules which help our immune system to recognize foreign substances. And these molecules are coded by certain genes, and those genes are together called as MHC complex, major histocompatibility complex. So in humans, the MHC is known as human leukocyte antigen. Why? Because most of the MHC molecules are seen on the surface of human WBCs or leukocytes. And they are called as the major transplantation antigens. And not only HLA system, the blood group antigens are also transplantation. So before doing transplantation, we always do a matching between the donor and recipient blood group antigens, and also the histocompatibility antigens. So if you get a properly matched donor with exact matching of the blood groups and also this HLAs, it will be very good acceptance of the transplant. So there is very less chance for rejection. But that's very difficult because HLA system, HLA proteins are they are very unique to different individuals. So if you want to get an exact matching, you'll have to go for an identical twin donor. If that's not possible, you have to get maximum matching. So that is the importance of human leukocyte antigens. So the HLA complex, they are the yellow antigens present on surface of human leukocytes. So you call it as HLA, and the set of genes coding for them is called as the HLA complex or the MHC, the same thing. And in which chromosome it is located, it's important. It's located on chromosome 6. So this is chromosome 6, and this has got 3 regions, that is A, B, C, which is coding for class 1, and the D region is coding for class 2. You have an additional region which is coding for class 3. Actually, MHC is mainly class 1 and class 2. Class 3 actually composed of certain complement molecules. They are coding for certain complement molecules and certain other molecules like factor B, all those which are involved in the complement system. So what we are going to see is just class 1 and class 2 MHC. So see, class 1 has got 3 regions, HLA A, B and C. Class 2 is in region D, which is again split into DP, DQ, DR. So QRS. So DP, DQ, DR for class 2, HLA A, B, C for class 1. So the class 1 antigen, that is HLA A, B, C, they are present on the surface of all nucleated cells. I repeat, all nucleated cells. So they are not present on RBCs, and they are also lacking in neurons. So they are present on all nucleated cells. They are the most important antigens involved in graft rejection and leading to cell-mediated cytolysis. So your cytotoxic T cells, CD8 positive killer T cells, they recognize the MHC class 1 antigens for their action. Okay, so class 1 is present on all cells. It is responsible for the graft rejection, and they are recognized by the cytotoxic T cells. Class 2, it's in the region PQ, PR, DQ, DR and TP, or you call it as HLA, DP, DQ, DR. They have limited distribution. Remember, class 2 is not as good as class 1. It's not present on all the cells. They are present only on the surface of specific cells which are called antigen presenting cells. So antigen presenting cells, those are the macrophages, dendritic cells and B lymphocytes. These 3 are the antigen presenting cells. This class 2 is present only on the surface of these 3 cells, and also some additional antigen presenting cells. So class 2 MHC is recognized by your CD4 T cell or helper T cell. So they are responsible for the graft versus host reaction, which is another kind of reaction taking place in transplant tissue. So more than that, this class 2 is very important in helper T cells, for helper T cells to recognize an antigen. So an antigen presenting cell process the antigen and show the antigen to the helper T cell along with the MHC class 2 molecule. So if you have a macrophage which has engulfed an antigen, this antigen will be processed by the macrophage and it will be expressed on the surface of this macrophage along with MHC class 2 molecule. So this is recognized by the T helper cell. So that is the importance of class 2 MHC molecules. So what is MHC restriction means? What we have already said, a CD8 cytotoxic T cell can recognize antigens only if it is presented with the MHC class 1 molecule. So with that, they go to kill the cells. They destroy the cells when they recognize the antigens along with MHC class 1 molecule, whereas CD4 T cells recognize antigens only when they see the antigen along with the class 2 MHC molecules on the surface of the antigen presenting cell. So this property of the T lymphocytes by which they recognize the antigen only with the specific MHC molecule. So you remember 4, 2 and 8, 1. CD8 MHC1 and CD4 MHC2. So that is what is called as MHC restriction. So that is about the MHC molecule. So please learn the complement pathways and the MHC HLA complex, which can be asked as short note questions for your exams. Thank you.